So I will repeat again. Okay. The major function of the Krebs cycle, okay, is to oxidize or remove electrons from carbohydrate, fats, and proteins to ready them for the ATP production. And as mentioned just now, it also called as a citric acid cycle or tricarboxylic cycle ataupun kita panggil sebagai PCA that involves 10 chemical reactions. Okay. So again, when glucose has been broken down into pyruvate, the pyruvate molecules are shuttled into one of many mitochondria. And once in the mitochondria, the pyruvate is converted to another important intermediate compound, which is called as a acetyl-CoA. And this process, a molecule of carbon dioxide is created, the first of 3 for each complete turn of the Krebs cycles. One can begin to see the production of carbon dioxide that goes along with the utilization of oxygen in anaerobic metabolism. The Krebs cycles is a series of 10 chemical reactions that begin with acetyl-CoA, a 2-carbon compound that join in with oxaloacetate, 4 carbons, to perform a 6-carbon compound. So citric acid cycle, citric acid, okay, what we call as a citric acid. So for this reason, the Krebs cycle is often referred to as a citric acid cycle. The rate limiting enzyme for this series of reaction is what we call isocitrate dehydrogenase or atau IDH. During the course of the series of reactions, 2 carbons are lost. That is due to the formation of 2 carbon dioxide molecules. And the reactions eventually return to the 4-carbon oxaloacetate, which is then available to combine with another acetyl-CoA, which we call as a repeating cycle of chemical reactions. Beginning with pyruvate, 6 oxidation-reduction reactions take place for each turn of the Krebs cycle. An aspect of chemical reactions that was not previously discussed, even though it occurs in glycolysis, is oxidation-reduction reaction. Some chemical compounds need to have electron removed in order to be transformed chemically to another compound. The process of giving up electron is called as a oxidation, and the compound that has electron removed has been oxidized. So the process of accepting electron is called reduction, and the compounds that receive the electron have been reduced. When electrons are removed, they can be destructive if allowed to remain free within the cells. Therefore, oxidation-reduction reactions are coupled. That is, when one compound needs to give up electron and is oxidized, another compound simultaneously accepts electron and is reduced. The two most important common electron accepting compound we call as a NAD and also FAD, which is nicotine amide adenine dinucleotide and also flavin adenine dinucleotide. Although most electrons are coupled in oxidation-reduction reaction, some can remain free within a cell and they may form damaging chemicals, what we call as a free radicals. Nanti kita akan tengok lepas ni what is mean of free radicals. So one of the Krebs cycles reactions is exogenic and releases enough energy to produce a high energy phosphate compound, which is guanosine triphosphate ataupun kita panggil sebagai GTP, which is then used to form an ATP. In 4 of the oxidation reduction reaction, NAD is the electron acceptor, and in 1, FAD is the electron receptor. The vitamin anisin, there is nicotinic acid and nicotinamide and riboflavin, a major component of NAD and FAD respectively. NAD and FAD shuttle the electrons to the final major component of oxidative phosphorylation, the electron transport chain. Okay. So actually what is a free radicals? Actually, electrons are traded between molecules in the course of oxidation-reduction reaction. So, dia akan berlaku traded, particularly in the Krebs cycle and the electron transport chain. In some instances, individual electrons may leak out the process leaving the molecule with an unpaired electron. This molecule, or we call as a fragment of molecule with unpaired electron, we call as a free radicals ataupun ROS, reactive oxygen species. The formation of small amount of free radicals is normal wherever oxidative phosphorylation or aerobic metabolism takes place. The concern about this free radicals formation is related to the damage that an excessive amount of these molecules can do, referred to what we call as oxidative stress. These ROS can damage cells in our body by inactivate the enzymes, breaking the DNA strand, binding the NAD, thus prevent it from assisting in another oxidation-reduction reaction, and also damage the fatty acid in cell membranes. The excessive oxidative stress has been associated with aging and the development of cancer, atherosclerosis, and other chronic diseases. Fortunately, our body actually has defense mechanism against the free radicals, including enzymes for removal and chemical that can react with the free radicals, which is antioxidants to provide a more stable molecule. Okay. So the last one we look at in terms of the electron transport chain. The electron transport chain is yet another series of chemical reactions that take place within the inner membrane of the mitochondria. The electrons are passed along a series of complexes containing compounds that shuttle the electron down the chain. Oxygen is the final electron acceptor, picking up electrons in the form of hydrogen to perform water. The rate limiting enzyme for the electron transport chain we know as a cytochrome oxidase ataupun CO. When the electron are deposited into the electron transport chain, the resulting series of reactions release enough energy to power the ATP synthesis reaction. When NAD is the electron carrier, enough energy is released to reform the 3 ATP, and when FAD is the electron carrier, the resulting ATP formation is 2. Okay, and there is a growing consensus based open recent research that these figures may slightly overestimate the ATP produced, and that the estimate may be closer to 2.5 or 1.5 ATP for NAD and FAD respectively. Although further studies are needed to confirm this view. Okay. In skeletal muscles, these electrons are transferred into the mitochondria via FAD, and other tissues use NAD to shuttle this electron into the mitochondria, which affects the final tally of ATP. The electron picked up by NAD in the glycolysis phase can be shuttled into the mitochondria as FADH and entered into the reactions of the electron transport chain. Okay. What we call in the glycolysis phase, 2 ATP, as you can remember, are used and 4 are produced, giving a net production of 2 ATP. In addition, 2 NADH are formed in glycolysis. In the Krebs cycle, if you can remember, 1 ATP is produced via GTP, and with 2 turns of the Krebs cycles for each glucose molecule, so 2 ATP are results. With the 2 turns of the Krebs cycle, 6 NADH are formed, giving a total of 8 NADH when combined with the 2 formed in the conversion of pyruvate to acetate. Using the traditional energy yield for NADH and FADH in the electron transport chain, 24 ATP are formed from the 8 NADH. With the 2 turns of the Krebs cycle, 2 FADH are formed. The 2 NADH from glycolysis are shuttled into skeletal muscle mitochondria as FADH, resulting in 4 total FADH and 8 additional ATP. Therefore, the net ATP for aerobic metabolism of glucose in skeletal muscles is 36 ATP, although the ATP yield may be as low as 32. In other tissues, the net ATP may be 38 if the starting point is glycogen rather than glucose. 1 ATP is saved, bringing the total ATP produced to 37 or 39 depending upon the type of the tissue. In comparison, the two other energy systems, this oxidative phosphorylation has significant advantages in that it can produce many more ATP and has a virtually limitless duration. However, the series of reactions are very slow compared to creatine phosphate ataupun anaerobic glycolysis and are dependent upon the provision of oxygen. Due to these limitations, oxidative phosphorylation is the predominant energy systems used at rest and also to support low to moderate intensity exercise. Example of exercise where oxidative phosphorylation is the predominant energy system include aerobic activities such as walking, jogging and distance running, cycling, which is longer distance and not sprint cycling, swimming, a longer distance, not sprint, and also dance aerobic. So this is you can see the graph to show the energy release over time. I mean from the ATP store, ATP PC, or what we call as a creatine phosphate system. This is a lactic system and this is the aerobic systems. Where the creatine phosphate can last to 5 to 10 seconds, the lactic acid system so 1 to 2 minutes, and aerobic systems can be longer, more than 2 hours. Again, before we go to the quiz, I have to make sure that to summarize the characteristic of the oxidative phosphorylation energy systems, we can go here. So it involves like 124 chemical steps or reactions. We have 30 compounds, 27 enzymes. They have what we call a rate limiting enzymes, which is IDH, isocitrate dehydrogenase for the Krebs cycles. They have a COX, cytochrome oxidase for the electron transport chain, and they have a PFK, phosphorylates for the anaerobic glycolysis. And this is a potentially what we call limitless duration. It is an aerobic system, and it's fatigue associated with fuel depletion, for example, muscle glycogen, and the predominant energy system endurance exercise, for example, long distance running.